Hi everyone, so I was just going to make this quick video to go over some of the resources that I posted and kind of to better prepare you to do the next, the phenology report and the lit review. And I just kind of wanted to take a more hands-on approach this time to some of the details that I'm looking for, especially in these specific sections. You have to remember that when you're writing a disciplinary scientific report, and this can be in science, this can be in healthcare, this can be in just about anything. It follows an objective format that nobody changes so that anybody can come in and interpret those results and basically apply them to the results that they are studying, for example. So we'll kind of go over each of these changes and how they can impact things. The dog is barking in the background if you can't tell. Sorry. Actually, no, she's okay. Okay, so for lit review, this is not meant to be an encyclopedia dump. It's meant to be kind of an exploratory, why is this species, or bacteria in our case, it's going to be tree with the phenology, why is this unique? What are the unique parts of this organism that are important to the scientific community, and how do you communicate that? And then in the final step with a lit review, and this is where I think a lot of us didn't take this final step, offer something new. Offer where, based on these results, what do you think is the next step for this organism? What do you think that there could be a use for it? Do you think that it's more or less vulnerable to disease, with the changing, let's say climate, economic landscape, anything like that? And that's something that you can get creative with. What's the next step in this? Or, even something that you would want to cover, what's missing. So remember that we're working in very small systems here because this is a Bio 102 lab. We still want you to practice and prepare for the time that you are looking at something unique that you want to affect change with, okay? Okay, so remember, this is your time to think critically and look at the next step. So I shared this with you, so you can always take a look at that. Okay, with the results, and we'll go over the example that I'm gonna post, you need to have a figure and table caption. That all needs to be complete. This is very important because, as I've told most of you, figures are probably the most important thing that you'll write because tons of people and tons of your readers are just going to look at the data in your figures. That's it. That's all they want to know. They want to, take the conclusions right there. If those figures are not clear or they're not very well done, the reader will immediately trash your paper and they will immediately think like, this is not serious. This is not good. Okay. So we'll have we'll have more data-driven figures in the next section. For phenology, because you're going to be measuring like the buds and everything, and you're going to see how those grow basically. But just remember, for figures, it's very important to make sure that they stand very nicely alone, and independently, so that any new reader is going to immediately know the point that you're coming across with. Okay. And then finally, the discussion is super. So figures are in results. For results text, just put out exactly what happened. It sounds like you're repeating, but there is a population of people, like I said, that just look at figures. There's a population of people that just look at the texts. And you need to have those readers, kind of taken care of because they want to see exactly what happened. The discussion is where you get more creative and kind of assign what is meaningful about your result. How exactly did you come to your conclusion? The evidence that you're making for essentially this court case conclusion. This is kind of your closing statement if you're in a court case, for example. Here's why my hypothesis was wrong or right. Here's the evidence that supports that claim. And here's where we go next. This is another chance to add, what's the next step for this research or what you would do. Again, we're working in a smaller system for what we're doing, but it's still a very good chance to practice that. That's what this education is you're supposed to be able to kind of create these opportunities for yourself to engage in higher thinking and higher, disciplines, and especially with disciplinary writing like this, that is eventually going to, you're eventually going to enter the big leagues with this. And we want you to be very, very prepared for that. And you should want the same. Okay, so let's go over an example right here. This is a class example from someone who I thought did, they did very well. And here, let's take a look here. There we go. So in this case, with the description, this wasn't a graded section, but this was a great, just basic practice on materials methods. Basically, what did we do? If somebody wanted to see exactly how to repeat this procedure, a materials methods section that's basically this is a perfect spot for that, okay? This has all the instructions necessary to, redo that section. Okay, so here's the results. Biggest thing, remember, we kept a lot of those plates, and that was kind of your prerogative if you wanted to include a picture. Some of you did, and I thought that was a really good move. Including a picture is very good. You can even have a small title of a picture in this case, so this isn't actual data figure, so you don't technically need a caption. since it's just an observation. Okay. So again, this is a good like intro to the results. Here is, kind of some of the morphological characteristics. And so this actually is doing a very good job, not serving so much as a caption, but it is a very good like large results text, and you are describing the results that we did see morphologically in this organism. So this is a very good section for that. Okay, next you can see that every result had its own table, and what that meant, the figures or the figure captions showcased what each result means. Positive test is shown here for bubbling, a negative result is shown no bubbling. Okay, so that goes in for your reader who is just reading for catalase tests. They're going to understand right away what exactly, what exactly happened in this test, what it proves, and exactly what we're looking at. So they do a good job following through on each of these. Each of these captions is detailed enough to obviously get the point across of what what happened in this test. Given that we didn't have a materials results section, it was fine if any of you included a lot more of the methods in here, so that's good. But again, each of these is essentially your figure. So we have tables in this lab, we'll have a figure or two in the phenology lab. Okay, same thing here and here. Okay, what we had here though is that, oh, that didn't work. Delete. Okay. What you had here, though, was a results text. Okay, you have figures, you have matching captions, but a lot of readers are going to look for the objective text. What exactly does it mean if you're negative or you're positive for these texts, and what exactly happened in our tests? Okay, now this doesn't have to be huge. This is just literally what happened, statement after statement. You don't have to get very creative with the language here. In a discussion is where you can start making some bigger interpretations. This is where they're going to cite specific results from their table to document basically the back and forth of what this species could be. Remember, we all did this in lab, and we all basically came to a conclusion based on the morphological characteristics and the table characteristics from what results were positive and negative. Okay, so that's where the discussion goes in, and it's basically like, here's what we got, here's our most likely genus in this case, is what we found. And this is why. And the evidence for our why and why we believe our hypothesis is correct lies in these texts, or in the in these results, and that's our evidence. So in this case, you can just effectively small conclusion about what what exactly this genus was. In this case, it was close to Pseudomonas based on the text, and basically how confident are you in this assertion. So in our case, we're pretty confident. We ran 5 tests. Are we exactly sure? Did we sequence the genome? Obviously not, but it's good to leave some ambiguity, or you have to measure how certain you are of this information at the end. So the discussion is where you can kind of get free and talk a lot more about why your results they do line up with your hypothesis or not. Okay, lit review is different. Lit review is where you're taking something and you're kind of doing a nice, basically not an encyclopedia, but what are the unique characteristics of this species in this case. So in this case, you can go over the basics, which is what family, what's their main role, things like that. Then you can start going into, in this case, how they do. You're going to start seeing, oh, that's going to go off the grid. You're going to start seeing how this leads to things like stem collapse, or how what things it infects. Because remember, you remember a lit review is still really focused based on the problem that you're looking at. It shouldn't just be the copy paste of a Wikipedia article. It should be unique characteristics of whatever you're examining based on the realm of what we're studying at that moment. Okay. So in this case, they cover disease. They actually cover how do you prevent infection. This was very good. So I mean, if you go into a healthcare scenario, it's usually, where does the disease come from? What are the symptoms? How do you treat it? How do you prevent it? Remember, those are 2 different things. So that's a good exercise in this if you're interested in healthcare. I know I probably overdo the healthcare side because that's kind of where I come from, but it's interesting. Okay, so you can conclude here basically what is special about it, and then right here, what are the implications of everything that you described all up here. Where do you think the future goes with this bacteria? What is the future research being done on it? What's something that you would implement possibly? It's a very creative section that I probably should have emphasized further for everybody. You definitely want to have one of these, because otherwise the lit review is just a 3 or 4 unique things about this bacteria. And so I really wanted you to go ahead and take a look into what's a special direction, what's something you can create in the next direction to, take the science forward on this, this thing. In this case, a certain type of bacteria. But remember, you're going to be writing these reports on things that are, very meaningful to you in the future, but you still need to have that writing practice so that the first time you present something like this, you're already ready for that next step. And that's going to be good. And that's what Gustavus is really preparing you for. Okay, lit review. Awesome. Primary sources, and we're going to go over what that means. Primary sources are going to be traditional research papers where data is collected. After a hypothesis was made, a question was asked, data is collected, you interpret the data, you make a conclusion. So in this case, these are experiments. These are full-on experiments. These are not just review articles. These are not encyclopedia articles. Okay, so that's why we had 5 there. That's what Nwang needed for us, and so that's, that was the point requirement, and we needed 5. So you are definitely allowed to use non-primary sources. We were just looking for you to use 5 for that practice to identify what is a primary source and what is not. So in this case, you're definitely allowed to cite non-primary sources. These are review papers, articles, things like that. But primary sources have actual research in them, and they are what drive citations because you're building on the science, or you are, reviewing the science in that case. Okay, so let's go here. This is a primary source. Okay, so usually the things to look for, there's a focused title of a question that you've asked. There are usually tons of authors. There's usually going to be an abstract, and it's going to detail that research was actually done in this case. Okay, in the abstract you should see the images of, we posed a question, this is why it's significant, here's what we did, here's how we did it, and what are our conclusions, okay? That's a big thing for primary lit, is that primary literature articles make a conclusion based on data that they collect, okay? So let's take a look at some other kind of telltale signs of a primary article. First of all, they're going to follow that simple format of intro, and it's going to introduce something. This is kind of like a literature review. That's what the intro is for. That's why we practice that. Next is materials methods, and there's going to be little subsections sometimes, but overall you're going to have this main section be here. Okay, so intro, materials methods, it's going to go through everything. You're going to start seeing results, okay? You're going to see tables, you're going to start to see figures. And then you get to the actual results text in a primary article, and each one is going to usually detail a single figure, okay? So in this case, specific DNA mutations are going to target stuff, and here we are. Here is figure 1. Okay, you can see that there's actual data. There are numbers. There are statistical tests running around. There are p-values. These are all dead giveaways that this is actual research. We're actually taking a look with data at questions that are unanswered, and we're going to try and answer them. Okay. So in this case, we are moving on. I think we are okay. I think we're stuck here. Okay, well, we'll just stop here. Okay, so if this is still recording, I think it is. Looks like my computer, I hope it's trying. Well, okay, I'm probably gonna end it here. So a review article is a secondary source. You're not gonna see the, intro, materials, methods, results section that you would normally see, and thus you're not gonna, see the actual data. That's what you need with a primary source is actual data. Review articles are going to have nice images. They may even have percents from collected data from other sources, but they're not going to have their own stuff. Okay. Sorry, my computer's old, so we'll call it at this. Look for primary sources for those reasons we looked. Secondary and tertiary sources are like articles, university websites, things like that. Okay. Okay. Email me if you have questions.